Hello everyone, my name is Kody Baumgardner and today I will be covering the topic of PacBio smart sequencing. So what exactly is smart sequencing and how was it developed? SMRT stands for Single Molecule Real-Time Sequencing. This method was originally developed in 2011 by Pacific Bioscience, which is an American biotechnology company based out of California. In 2018, Pacific Bioscience entered into a purchase agreement with Illumina worth approximately $1.2 billion. Now, smart sequencing is a next-gen method of DNA sequencing which is capable of providing long base pair reads, a uniform coverage of a sequence of interest, and can provide epigenetic information of a specific sequence. Now, how exactly is this method of sequencing performed? The first step is to develop smart bell libraries from one's isolated template DNA of interest. This is done with a proprietary kit in order to remove damaged DNA, create a circularized DNA template, and prepare the substrate by annealing a primer to the genomic sequence. The DNA is then loaded onto something called a SMRT cell, which contains approximately 8 million zero-mode waveguides. These zero-mode waveguides are small pits with a mobilized DNA polymerase in the bottom. DNA becomes trapped within these pits, and then fluorophore-linked nucleotides are added to the sequence reaction. With smart sequencing, the addition of nucleotides can be tracked in real time as they are added to the target sequence. Once the template DNA is loaded onto the SMRT cell, it can then be placed into a bioanalyzer for sequencing. The addition of fluorophore-linked nucleotides creates a fluorescent pulse when binding to the DNA sequence. The guanine, adenine, thymine, and cytosine bases each have different colored fluorophores attached and release individualized light pulses, which are then directed by the zero-mode waveguide into the sequencing machine. The sequencer notates each individual fluorescent pulse and creates a movie in which sequence information can then be extracted. In the past, the SMRT cells with loaded template DNA were placed into the PacBio RS2 for analysis. The RS2 provided read lengths on average of 10,000 kilobases and produced approximately 1 gigabase of raw data per SMRT cell. As of 2015, Pacific Bioscience has developed a new sequence system named the Sequel. The Sequel has improved read lengths ranging from 30 to 50 kilobases and improved throughput of approximately 20 gigabases per SMRT cell. So what exactly are the advantages of using smart sequencing for genetic analysis? One major advantage is the amount of time it takes to perform a sequencing run. Run times are typically 30 minutes per cell, and a total of 8 cells can be run at a time, leading to a maximum run time of 4 hours. This comparatively short runtime could be crucial when using the smart sequencing system for diagnostic purposes during epidemiological outbreaks. Also, the long read lengths provided by smart sequencing are great for constructing de novo genetic information. When compared to short read lengths, long reads can span repetitive sequencing regions and provide increased coverage for difficult locations within a genome. Additionally, smart sequencing can provide information on DNA methylation, which is important to understand how locations in a genome are regulated. As fluorophore-linked nucleotides are added, if a region within a sequence is methylated, an altered fluorescent pulse is produced, which a smart sequencer can detect and provide epigenetic information. While smart sequencing can provide notable benefits in certain aspects, it also possesses numerous disadvantages as well. Typical smart sequencing provides a relatively low output of 500 megabases to 1 gigabase per cell, making the system less optimal for whole genome sequencing. Smart sequencing also tends to be more expensive than other methods, with costs ranging anywhere from $1,500 to $3,000 per run. Additionally, the price of PacBio Sequel analyzers is around $750,000. There's also a high error rate associated, ranging from 10 to 14% of all reads. Fluorophore-linked nucleotides can become trapped within zero-mode waveguide pits, leading to misreads of the sequence. When comparing smart sequencing to Illumina, Illumina sequencing provides much smaller reads between 150 to 200 base pairs. Illumina tends to be much more cost-effective, with prices ranging from $50 to $150 per gigabase of genetic information. Illumina sequencing does take a considerably longer time to run, with analysis taking about 27 hours to complete. One major advantage Illumina has over SMRT sequencing is the error rate, which is approximately 0.1% per run. SMRT sequencing has been a major benefit to the scientific community since its conception, providing genetic sequencing information on over 1,000 papers in 2019 alone, being published in prestigious journals such as the New England Journal of Medicine, the Journal of Immunology and Cell Biology, and Nature Biotechnology. SMRT sequencing has been used to discover genetic variations in prevalent diseases, determining the genetic diversity amongst immunoglobulin heavy chains within the human body, and for the identification of bacterial species by analyzing DNA methylation. Thank you again for your time, and I hope that this presentation has been informative.